A gene drive is an example of one of those techniques that falls into the just because we can, should we category. It is a quite alarming technique as it introduces changes to an organism's genome that are self-propagating and non-Mendelian. Gene drives have potential uses in eradicating mosquito-borne diseases such as malaria and Zika, and tackling drug-resistant human fungal pathogens and eradicating invasive species. So what is it and how does it work? Okay, usually when you introduce a homozygous mutation into an organism, at the first cross, assuming the other organism is wild type, you get heterozygous offspring. Effectively, over time, the mutation may dilute out unless it gives a selection advantage. However, if the homozygous mutant were a gene drive mutant, then the offspring would be homozygous for the mutation. But how can that be? That is not Mendelian. Well, the gene drive approach uses, you guessed it, CRISPR. And in a gene drive, you introduce not only the mutation, but also the CRISPR machinery that can propagate the mutation to the other chromosome, hence making the heterozygous organism homozygous. That is, you drive the gene through the population. If the organism has a mutation package, it will be homozygous.